Hey everybody, it's Mr. Dan, and in this lesson I'm going to talk to you about translation or protein synthesis. Okay? So this is part 2 of our lesson about transcription and translation. So here, as we get started, let's take a look at just this scene that we've got going on in this cell. This is a eukaryotic cell, which means it comes from either a plant or an animal. And it's really easy to tell if you have a eukaryotic cell because it has what we call a nucleus in it. And that's where all the genetic material is stored. Not all cells have a nucleus in them. Bacteria cells, for example, or prokaryotic cells, they don't have a nucleus, and they just kind of have their DNA everywhere. We'll take a look at that a little bit closer here in just a second. But what I want to do before we get going here is just take a look at this picture. This is showing us the whole chain of events from DNA to RNA to protein. And that's what I really want you guys to have mastered. I really want you to be able to describe everything that's going on in this chain of events here. So let's quickly just go back and review that, right? So we're talking about how DNA turns into RNA, which turns into a protein. And then ultimately, the combination of proteins that a cell has is going to determine what that cell does. So not all cells in an organism have the same proteins. They have all the same DNA, but they don't all have all the same proteins active. So the combination of those proteins is going to determine what it does. A nerve cell has different proteins active in it than a muscle cell or maybe like a skin cell. They all have different things that they've got to do. So they need different proteins. Proteins are the little machines inside of a cell. They're molecules that do all the work of the cell. There's nothing else in the cell that actually does anything. Okay, so here's a eukaryotic cell. This is an E. coli cell. It's bacteria, and it's really easy to tell because there is no nucleus in this. We have all the DNA, but it's kind of just stuffed in the middle. And then we see these ribosomes around the outside. Okay, so just to review this, we talked about the structure of DNA and how we have all the nucleotides on the inside. That's where the information is stored. We use RNA polymerase, this molecule here and here, to unwind the double-stranded DNA and then to make a copy of the DNA onto what's called mRNA. Okay? And so this happens one nucleotide at a time. The RNA polymerase is able to open the double-stranded DNA, is able to move along that DNA, and it's able to make a copy of it at the same time. So this is a multifunctional enzyme that we're working with here. And then we looked at ribosomes, and these are really big protein complexes as well. And the job here is to take that mRNA and turn it into a protein. And when you look at the structure of it here, you can see that there's protein parts, the blue or the yellow, the blue, the green, down here below this other subunit. These spirals, these alpha helices going around it. These are all different protein structures within the ribosome. And it has what's called rRNA, or ribosomal RNA, attached to it also. And so this big molecule here, its job is to turn mRNA into a protein. So it kind of makes sense that it would have both of those parts to it. All right, so that's a ribosome, a couple different looks at it. Here's a simplified view of it, and we see the RNA going through the ribosome and out coming the new protein. So what we want to address today is these two big questions of how does a ribosome actually build the protein? It's more complicated than this picture here. And how does it know how to do that? So back to this scene here where we see the big scaping view of everything that's going on here. And we're going to start right here with the ribosome reading this mRNA and then out coming the new protein. So the way this works is we start with a piece of mRNA, and this is a single-stranded nucleic acid. It has a 5' end and a 3' end. And what's going to happen here is the mRNA and the ribosome are going to attach to each other, and the mRNA is going to sort of like feed through or be pulled through the ribosome, almost like you're pulling like a string through it. It's going to go in a specific direction. Really what's going to happen is the ribosome is going to move along the RNA. So here we see the translation direction. What this means is that the ribosome is going to be moving from left to right across the screen. Okay? And I can tell that right here because I kind of know how to read this. The way the ribosome reads this is it looks at the sequence of letters or nucleotides that's on the RNA. There's 4 different nucleotides, and they're in a big long chain in some order. And the way it reads it is 3 letters at a time. Every 3 letters here is what we call a codon. Okay? So 3 consecutive nucleotides that correspond to a specific amino acid. Okay, so or 3 letters in a row that match to an amino acid. So there's a chart that goes along with this. These are the 2 most common ways to see it. We've got a picture like this in the classroom. We've got a picture like this in one of the modeling kits that we do. But here you can see all the different 3-letter combinations that are possible and the amino acids that match up to it. So there's 20 amino acids. There's 64 different combinations, but there's 20 amino acids. Okay, so some of them double or maybe triple or quadruple up in some of these cases. Okay, so these are the combinations that the ribosome is going to be looking for to read. And it's going to match an amino acid with each one of these combinations. So take a look right here. This one, AUG, is what we call the start codon. Okay? It's methionine is the specific amino acid. But that's the first one that the ribosome is going to be looking for. So here I have it highlighted, AUG. That's the start codon. And so that's where the ribosome is going to start reading. Anything else that was on the RNA before that, it just kind of ignores it. But then we get to this start codon, and that's where it starts doing its real work. So what happens here is when we start getting these, now every 3 letters becomes a new codon. So AUG, that's the start. UUC, that's another one. AAA, that's another one. And we could go back to the chart and look at that. So here, UUC. What's that going to be? Well, we kind of look. Here's UUC. Here we go. So that's phenylalanine. That's the amino acid that's going to get attached. AAA. Let's see here. AAA. Lysine is the amino acid that's going to get attached here. And look, that's what they have in this chart. Lysine, which matches up with the AAA. So this is how we match the amino acids with it. But how does this actually happen? There has to be some moving parts, some functional parts to make this happen. The ribosome isn't just snatching these things out of the sky. What's happening here is that there's another type of RNA, right? So far we've covered mRNA, which is what gets copied from the tRNA. The mRNA, which gets copied from the DNA. There's rRNA, which is a part of a ribosome. And then there's this tRNA, and that stands for transfer. And so what's being transferred here are amino acids. See these funny shaped things here? These are floating around. These are the tRNAs, and these are floating around in the cytoplasm of the cell. So just like inside the cell, these things are floating around all over the place. There's billions of these things, okay? There's also all of these amino acids floating around in the cell. Again, billions of these things. There's so many of these. They're everywhere, okay? And so what happens is that there's a specific tRNA for every codon. And here you can see how it matches up, right? A's and U's, U's and A's, C's and G's. So these all match up on one side, and for this particular set of letters on the tRNA, there is an amino acid that matches with it, that will bond with it. So that's what we see over here. Okay? So these things are floating around. They're everywhere inside the cell. And when this ribosome starts to translate the code, it matches the right tRNA with the code on the mRNA, as we see here. And once that happens, it sort of lets go of its amino acid and floats off into the cell to go find a new amino acid to match with it so it can come back again. So these things are everywhere. They're just waiting to come in and have their name called so they can bond with the mRNA and the ribosome, donate its or give off its amino acid, and then detach and go find a new one. Okay? So it's kind of a crazy process going on in there, but this is all happening. It's happening really, really fast. Watch that video earlier in the slideshow and you'll see what I mean. Okay, so I think I said everything we need to say about that. Now, if we go back here, what's going to happen as a result of that is that we're going to end up with a chain of amino acids like we see here, okay? And so this is what we call the primary structure. This is just the sequence or the order of amino acids as it comes out of the ribosome. And you can think of it as just like a straight line. It might be kind of wiggly or something like that, but it's just the primary structure just means it's the order or the sequence of amino acids that's coming out of the ribosome. So proteins, we talked about, their shape is what determines what they do. So this sequence here is going to start to bend and fold and twist into different shapes. So you have secondary structures, which are these alpha helices or a helix, the twistiness to it. There's beta sheets, which kind of make a zigzag shape. Okay, so it's going to start forming into those, and those are going to form into a whole complex that we call the tertiary structure. So this is the folded structure of the protein, and it's what you're doing in that amino acid starter kit where you build the protein molecule. Okay, so this has a very special, very specific shape. The shape is determined by the order of amino acids that are built by the protein, or that are built by the ribosome, which is built by the RNA and determined by the sequence of the DNA. So DNA turns into RNA, which turns into protein, and then that protein folds to make a special shape, which has a special job. Here's what we call a quaternary structure, and what happens here is we have multiple proteins that are coming together to create a bigger complex. Okay, structure equals function. That's the point here. And I want to give you an example of this. Okay, so here's just another look at this. This is a specific amino or a specific protein here. This is insulin. So this is like what we're working on. Another one that we're doing in class. Insulin is two separate chains of amino acids that fold and combine with each other to make a single protein. So here we can see the two different chains based off of the colors. You can see some of the secondary structures like the alpha helix here. Here's another one. Here's another one. Here's what we call a beta sheet, this zigzag thing here. It has these secondary structures that form its tertiary structure like we have here. Okay, so what I want to talk about is how the structure of this, if we change it, it's going to change the actual job that it does. So insulin is a protein, and its job is to break down glucose in your bloodstream. People who have diabetes don't make enough insulin. Okay? So the way this structure works is your body makes the insulin, right? It makes this down here. And then this gets released into your bloodstream, and it starts to metabolize or break down glucose. So it makes it one protein at a time, but these proteins combine with others to make what you call a dimer right here, and then 3 dimers come together to make what's called a hexamer. Okay, so don't get too worried about this, but basically what we have is 6 insulin molecules that are all stuck together in one big structure. So this is what the cell makes, and then this gets released into your blood. The way the protein works is once it goes into the blood, this whole thing starts to break apart into smaller pieces, and then those smaller pieces go find glucose and metabolize the glucose. People have different forms of diabetes. Some people have it really strong. Some people have more of a mild case. So some people need insulin, right? They take a shot of insulin and they need it to act really, really fast. And so something that a chemical engineer might want to do, a biochemical engineer, is they might want to make this molecule so that it breaks apart even faster. I did a study, I read an article about a form of this protein where it's actually meant to break apart really slow. And so what they did in that is they took one single amino acid and they changed it to a different one. And what that did is it created a stronger interaction between these two proteins here. So the whole thing held together longer. As a result of that, the whole complex held together longer, which meant that the drug would last longer in people's bloodstreams. So because it lasted longer, someone who had diabetes could take a shot maybe once a day or even once a week, and it would still be effective in their system. So there can be a real functional change by making a small change to the structure, right? This affects how people take their medicine. If you have diabetes, you don't want to take a shot every day or maybe after every single meal. Or maybe you have a really strong case of it and you need it to work really, really fast. Well, we can make that happen by making a small change to maybe just one amino acid here. Change this one for something else, and it's going to make a small change to how the rest of this molecule works. So I tell you this story because it's important to think about how does the structure of DNA affect the structure of the protein and the structure and the function of it and what it's doing inside of cells. So one last thing to talk about when we're trying to discuss what's going on inside the cell. Take a look at this picture here. It looks kind of like some UFOs or something weird going on. But what this picture here shows is that we've got a stretch of mRNA, and we actually have multiple ribosomes attached to it. Okay? And then we have the proteins coming out of it. So what's happening here is we're making multiple proteins off of the same mRNA at the same time. Okay? This is going to speed up the process and make the cell a lot more efficient. Okay? Our bodies need to make proteins really, really fast sometimes. Like after you eat food and you need to make some insulin. Okay? That glucose in your body, it's actually toxic and you don't want it to be in your bloodstream very long. So your body has to break it down immediately. Here we see the DNA, and it has multiple transcription sites. And within those, each transcription site, there's multiple ribosomal translation sites. Again, showing us how quickly our bodies can be. It's not just a one-at-a-time thing. It's happening in multiple places at multiple times. And so here in this situation, I count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 24 different sites. I mean, this is made up here, but it's probably more than that, okay, for how many times we can make protein. So each one of these is going to make its own protein. Okay, so that's just a little bit of insight to how the body's going to do this. All right, so back to this big view, right? DNA to RNA to protein through the whole process. Transcription inside the cell, happening at multiple sites by polymerase. That mRNA leaves the nucleus, and it's going to find all sorts of different ribosomes here where it's going to be doing translation and then folding into its final structure where it's going to go do the work inside of the cell. Okay, so that's the whole picture for us. I hope this video is helpful for you guys. If you have any questions about it, please let me know. Put them in comments, ask me in class, and I'll be ready to answer you. All right? See you in class, guys. Thanks.